Thank you, Dustin, and I'm going to turn it over to Dr. Dage.
I'll try and get started fast too. Not that fast. So I'm going to talk to you about biofluid biomarkers to aid in Alzheimer's disease diagnosis and drug development. Bruce gave a great introduction. So I did spend a long time at Eli Lilly, and much of the work that you'll see here actually originated out of the discovery work there. I do want to have a disclosure slide because I have a lot of disclosures. I'm involved in a lot of different things, including patents that relate to the phospho-tau measures. They're all assigned to Eli Lilly, so I have no benefit, but it's on there. As well as grant support and research support from different companies. So biomarkers for Alzheimer's disease pathology, it's really about amyloid, as Dustin mentioned, and that can be measured with CSF, A beta 42 peptides and 40 ratio typically, and that can be measured in cerebral spinal fluid or plasma. The plasma breakthrough really came around 2017, I think, with a couple of papers from Randy Bateman and a group, a group out of Shimatsu in Japan. But then neuroimaging, amyloid PET, which Dustin showed some data, is really heavily used in clinical trials, especially in the United States. The United States heavily relies on amyloid PET in clinical trials. They use CSF only when people are contraindicated for PET or don't want PET. It's not the same in Europe. In Europe, it's cerebral spinal fluid is more routine and often and goes without any problems. It's really thought to be pretty non-invasive. So keep that in mind if you want to participate in research. So tau is a little bit newer. The tau PET tracer is just only recently approved by the FDA, but the tau PET tracer, can measure insoluble tau, intracellular insoluble tau in living people. But then we also have a plasma phospho-tau measure, and we have a whole bunch of these, and I'll talk a little bit more about that. But those are all research use only. Nothing has advanced to FDA approval. They are just now becoming available as lab-developed tests so that they can be ordered by a physician. And I'll mention more about that later. And then neurodegeneration, it's not really AD specific, right? This is just really general. And we heard earlier about this using neurodegeneration for sports concussive injury. And so you can look at astrocytic markers like GFAP or S100 beta. In this case, I think I have neurofilament light chain, which is used really heavily, and we saw that earlier as well. But it is very non-specific. And in fact, in Alzheimer's disease isn't one of the diseases where you see the largest changes in neurofilament. But it is elevated, and it is very useful. And I'll talk about that later too. We'll see how much later. But first, that's the basics of Alzheimer's disease pathologies: amyloid, tau, and then a little bit of neurodegeneration. But that's not it, right? Alzheimer's disease has a lot of different components, and there's a lot of different measures, whether it be in cerebral spinal fluid or plasma. And they can be associated with astrocytes, which I mentioned already, and GFAP, which we can measure in CSF or in serum or plasma. Interestingly, the data is a little less correlative between CSF and plasma GFAP, which is of interest all on its own. But then there's also measures for microglia, so we can measure soluble TREM2 in CSF. There's a lot of neurodegeneration markers. A lot of them are synaptic, and so we call them synaptic markers, but maybe they're just general neurodegeneration markers. We don't know. We have a lot more work to do and try and figure that out. But there's all kinds of measures, and there's always new measures. There's lipidomics, there's proteomics in CSF and in plasma, and all these things are coming really fast, especially since we've had breakthroughs in measuring 42/40 in blood in 2017 and then phospho-tau in 2018. And so I'm going to focus really now on phospho-tau. That's my favorite, and I already gave you my conflicts of interest, so keep that in mind. But I wanted to talk about it because it's so very complicated. And we heard earlier about big tau, right? We heard about big tau and how it might be informative and not related. And that was one of the things that we did at Lilly, is we developed an antibody that wouldn't bind to big tau. And why did we do that? Because it wasn't really related to the dementias, right? And so it cleans up our signal and it gives our assays a little bit better performance in separating normals from disease. And that was one of the things we found and we thought was interesting and we implemented right away. So our assays, all of the assays that I developed are really measuring the 6 brain dominant isoforms. And that's, that's become more in vogue, I guess now. People call it brain-derived tau. Well, all of our assays always were that, but we didn't market it. Hindsight 2020, I guess. But so this slide really is just showing you there's multiple phosphorylation sites, 181, 217, 231. There's lots of them. There's many, many more. We heard about them this morning. But those are the 3 dominant ones that people are measuring in blood. On the left-hand side, you see a lot of the assay designs. When you hear someone say, oh, I measured 181, well, there's so many different ways to measure it. And all of these assays have subtle differences. Anytime you have a slightly different epitope, you can get different binding properties. You'll have different interference. We even heard about, the tyrosine modifications that, I think it's NY29, or it's, phospho 29. It's also nitrosylated. And it's interesting to think about those competition post-translational modifications. But there's so many different things and so many different details about assay design that you can't really just read a paper and take it for granted that it's going to be the same as the next paper you read. And so it's really important to pay attention to these details if you want to see that the assay is robust, if the findings are consistent. And I can tell you that if you look at the studies where it has the assays that we developed and that we implemented, they are consistent, they are robust. The cut points can go from one study to another. And so although the field looks confusing, the actual science, if you look at the details, it's actually really robust. And this just shows a whole bunch of different ones. And you can see in CSF and in plasma, the performance is really high with high diagnostic accuracy on the right-hand side. And what I mean by that is the area under the receiver operating characteristics curve is near 0.9 in both CSF and plasma. And so how did we get there? I mentioned this, like, the 40-40 ratio in blood was really, thought to be a big breakthrough around 2017-ish. In 2012, a lot of people, including myself, were measuring just tau. We were measuring tau in blood and thinking, oh, this is cool. Look, it's different. Statistically, there was a lot of overlap. It was statistically significant, but not clinically useful. And so what I'm showing on the left is a tau assay that I developed while I was at Lilly that takes into account, again, that's selective binding antibody. And that data is actually the best tau measure that you'll probably find in blood. All the other ones can be compared to it, but this one will always give you the largest effect size. And so you see that it's 36% is the mean difference. And so when you see in the individual dots, you see a lot of overlap. And that's what I mean by clinical utility, right? If you get a result, what does it mean for a person? It's great that it's statistically different, but what does it mean for a person? That's important diagnostically. And so in 2017 to 2016, and we published some of these papers and some of these findings with tau, with Michelle Melky at the Mayo Clinic, but then we realized that we needed a better assay, and that's where we started looking at this measurement of phospho-tau 181, where we developed some ultra-sensitive ways of measuring it with some really high affinity antibodies. And now you see that effect size goes from just a few percent, 36%, it goes up to 159%. Well, now you have a bigger mean difference. If the variability is low, you have a bigger signal window, bigger effect size. Now you can start to see that, hey, this actually could be useful. We could have a cut point and have people below a cut point, people above a cut point, and now they're talking about an intermediate zone, right? And so you can think about how you could use this and inform on the presence of Alzheimer's disease pathologies. And now it's a phospho-tau measure, and in this case, it's showing you an ADCSF profile, so that's amyloid and tau. But it wasn't good enough, and so we kept working to identify a better assay, and it was all about driving the effect size. And so when I talk about better assay, the design, the selection of the antibodies, what we targeted, all went after driving off of an effect size. And you see that with our phospho-217 assay, now we're up over twofold, so we're 227%. That's way better than where we started. but it wasn't some magic thing that just happened all of a sudden by serendipity. It was a lot of hard work, planning, science all along the way, and we were really driving for improvements all along. And that's where we are now. And in this 217 assay, and this is a paper from just I think it's this year it came out. It was online maybe at the end of last year. But this assay, this 217 data first came out in the JAMA article, and I think it was 2020, 2021, maybe 2020. But if you look at all of the papers that have that data, that assay is rock solid and consistent over multiple cohorts. In the YCAP study in New York City, a diversity study, it's rock solid. There are impacts. So when you have comorbidities, so patients who have large numbers of comorbidities as they get older, that's a normal thing. As you get older, you accumulate disease. Well, some of those comorbidities can affect the levels of biomarkers in blood. It's not too surprising. Chronic kidney disease, it impairs protein clearance. You're going to have more proteins in your blood. So a lot of these things are very intuitive. But as you interpret biomarkers, you have to keep that in mind and think about it. So I wanted to move on, and because I talked a little bit about, at the beginning or a couple of slides ago, about diagnostic accuracy. So I thought I'd come back to that and show this. So this is data from a collaboration we did with UCSF, Adam Boxer's group. And I wanted to point out on the far left, it's a receiver operating characteristics curve and showing a very, very high AUC of Alzheimer's disease against unaffected controls. Well, no surprise, like I'm sure Dustin can tell pretty fast the difference, right, from a clinical perspective. And so this is interesting, but it's not really that informative because that's not how we want to use the test. It's really about the AD syndrome versus the FTLD syndrome. Now here's two patients, both are impaired. Maybe you want a little bit of help to determine what diagnosis is most in line with what's going on. And so you can look at a lot of the testing and the cognitive testing, neuropsychological testing, that gives you a big hint. But having a test that then confirms the pathology that's present or absent could be a real big advantage. And that's what's shown here. And you see the area under the curves are, for both tests, are above 0.9, with p-tau217 being at 0.93. And what that means is shown on the right side is that four-fold effect size now, right? Now this is a memory clinic population where they're seeing a lot of different patients, but you see a really large effect size. They're color-coded based on PET scan, and this is really important because you'll see that some of these conditions like cortical basal syndrome or the CBS or progressive supranuclear palsy, which is the PSP, if you look at that and you see some of those dots that have elevated phospho-tau for those diseases, it's because they have coincident Alzheimer's pathology with that diagnosis. So this is a comorbidity, right? And so you have to think about, well, now, how do we know what's the driver? What's driving the disease? So if these patients were screened and identified as amyloid positive, they become eligible for lecanemab treatment, is it gonna work or not? You know, there's no studies to say that it is. So I think we have to do a lot of work to understand the effect of these co-pathologies and things that are presenting differently than just straightforward Alzheimer's disease, kind of what Dustin was talking about with early onset. Boy, it's surprising how slow I'm going through my slides. I better talk less. In 2018, they came out with this NIA research framework, and this kind of pulls it together to kind of illustrate how you use biomarkers with cognitive stage. And so I was talking about this when you look at two symptomatic people, but now think about, well, what if I want to look at people who are just perfectly normal, but they just happen to be 65 years old, and kind of like measuring cholesterol, we want to see what's going on in your brain pathology. So that's what this cognitively unimpaired, and you can see if you're showing up as phospho-tau positive, you have what they're calling preclinical Alzheimer's disease in this chart. And so that means you have no symptoms, but you have a high risk of having the pathology in your brain. Now, what does that mean? Well, we've done a lot of studies to follow people longitudinally, and it means you're at an increased risk of progression to MCI or dementia. And so it does mean something to know, but then there's currently studies underway with these amyloid removal therapies in this population to prevent the symptoms from ever happening, hopefully. You know, that's the wish, that's the hypothesis. We'll see how those trials turn out, But using these blood tests allow you to do those studies. Those studies weren't possible before we had the blood test to enable this kind of screening, because if you do PET scans in 30,000 people in order to enroll 3,000, it costs you hundreds of millions of dollars, and it takes way too long. And so it just wasn't feasible. And now a blood test makes it feasible. So it really is a huge impact to drug discovery. And so, gosh, I'm already out of time. So I'm going to finish with the diagnostic part. So this is really about the use of the blood test in diagnosis. And there's different ways of engaging. So I talked about screening, right? That's on the far left side of this, screening in healthy people. That's not really recommended right now, right? Because we don't know what it all means. We don't know really a lot of the details about the impacts of comorbidities. And, is it really going to be the driver that your physician wants to treat? Or do you have other diseases that are more important or more pressing to take care of? And so it becomes this prioritization of different things for an individual patient. But it's something that's on the horizon, and we're going to be continuing to do a lot of research in it. But what is being used are the more red colored ones, and that's in clinical assessment and evaluating the biomarkers in the course of diagnosis. And that's just like CSF is used. So CSF is an FDA approved test. Blood tests are not, it's research use only or lab developed tests. But they're going that direction. And with the performance of those tests of measuring phospho-tau in blood is now, with the improved tests, are reaching the same level of confidence or diagnostic accuracy as the CSF test. So it really is pretty exciting. This is drug discovery. There's a lot of different parts of drug discovery. So the use in clinical trials, I mentioned this already in the preclinical part. I'm going to jump into this. So this is how the screening happens. I mentioned this already, so I won't go into a lot of detail. But basically, you can screen 30,000 people with a blood test, which is pretty fast and simple and less expensive. It still costs you $1 million maybe, but it's not hundreds of millions. But then what you come out of that with, based on these criteria on the left-hand side, is you get people who have a greater than 90% chance of having amyloid and tau pathology. It's only 50% by tau PET, but it's 90% by pathology by IHC. And then these can be used, this blood test can be used in clinical trials as pharmacodynamic endpoints. And what's really important here is these blood tests allow you to change the way we do Alzheimer's research in the future, clinical research. Because now you can look at this and you can say it's a 18-month study. We have this biomarker that shows us a good effect in only 24 weeks or 3 months. Well, it's not 3 months, right? So however many weeks that is, 6 months. So it shows you a real robust effect in just 6 months on only 100 people per arm. So now if you're doing proof of concept studies, rather than spend this much money, that I won't tell you how much it costs, but we could probably do 10 studies at that shorter time frame and for this smaller number. So now we can test way more compounds, which will advance therapeutics that much faster. And that's all because of these blood tests and these measures are becoming available. And finally, just to thank everybody, there was a lot of people involved, as I mentioned already. And certainly I've only been at IU School of Medicine for 2 years, but everyone there is awesome and has been very welcoming.